Welcome to Night Shift Knowledge, a quiet place in the late hours where you can finally let go of the day. Take a moment to settle in, soften your shoulders, and allow the busy world to fade completely into the background. Tonight, we are going on a gentle journey through the fascinating corners of science, biology, and history. Before we begin, please know that the screen will fade to dark very soon. You can safely rest your eyes and leave this video playing as you drift off to sleep. I would also love to hear from you. Please leave a comment below with a general topic you would like me to cover in a future video. It is always a pleasure to read your ideas and shape this channel around what brings you peace. If you find comfort in this space, a like and a subscribe would be deeply appreciated. To ensure the next video quietly finds its way to you, please tap the notification bell. So as your breathing slows and your room grows a little darker, let your curiosity drift along with my voice. Let us begin. The mystery of the electric mud. The red LED flickers once, then steadies into a dull, electronic hum. There is no battery pack here, no solar cells, no hidden wall outlet. Just a jar of sludge pulled from a riverbank and a circuit completed by the Earth itself. It looks like a magic trick, a biological anomaly that shouldn't exist. We are taught that dirt is an insulator, something that stops the flow of energy. Yet here the mud is conducting. It is powering a light. The secret isn't in the dirt, or the water, or the metals dissolved in the solution. The secret is alive. It is a hidden world of microbes that have evolved a power source alien to our own understanding. To understand why this mud glows, we have to leave the laboratory and travel back to a time when we thought we knew everything about how life survives. The year is 1987. The setting is the Potomac River, a waterway usually associated with politics and history, not microbiological revolution. Dr. Derek Lovely, a young microbiologist, wasn't looking for electricity. He was looking for life. Specifically, life that shouldn't be able to breathe. On the surface of the Earth, oxygen is king. It's the high-octane fuel that most organisms, including us, burn to stay alive. But deep in the river mud, oxygen is scarce. It vanishes within millimeters of the surface. Down here, in the dark, suffocating sludge, life has to improvise. Lovely knew that bacteria in these depths survived by breathing minerals instead of gas. It was a process called anaerobic respiration. The prevailing wisdom was simple. These bacteria ate organic matter and exhaled by dumping their extra electrons onto nearby minerals like iron or manganese. It was a chemical exchange, a closed loop. But when Lovely isolated a new strain of bacteria from the Potomac mud, he noticed something peculiar. He named it Shewanella. These bacteria weren't just interacting with the iron when they happened to bump into it. They were consuming it at a rate that defied the laws of chemistry as they were understood. In the lab, Lovely watched in disbelief as the bacteria ate electrons faster than they could physically touch the iron source. It was as if a person were eating an apple while standing across the room. How do you breathe something you can't touch? How do you exhale an electron onto a rock that is centimeters away from your cell wall? The answer required a complete rethink of biological engineering. Lovely hypothesized that these organisms were physically reaching out into the world. He proposed they were growing biological appendages, tiny conductive filaments, to bridge the gap. He called them nanowires. If true, these were the world's smallest electrical cables, grown by living cells to connect to the mineral grid of the Earth. It was a stunning evolutionary breakthrough. Life wasn't just adapting to its environment, it was rewiring itself to become part of the electrical grid of the planet. But Lovely's claim hit a wall of skepticism, a wall built on the fundamental properties of biology. In our world, conductors are usually metals: copper, silver, gold. Insulators are biological materials: wood, skin, bone, hair. Proteins, the building blocks of all life, are notoriously bad at conducting electricity. They are wrapped in water and fatty membranes, nature's perfect insulators. The idea that a biological protein could carry a current over long distances was, to many physicists, a biological paradox. The resistance should have been too high. The heat should have destroyed the cell. The structure simply shouldn't work. The scientific community demanded proof. It wasn't enough to show that bacteria could make a light blink in a jar of mud. They needed to see the wire. They needed to measure the current flowing through a single living protein strand. The debate raged for years. Was the electricity real, or was it just a chemical illusion caused by the mud? To settle the score, researchers would have to descend deeper than ever before, past the cell wall, past the membrane, into the atomic machinery of life itself. Anatomy of a living wire. It is a light that shouldn't exist. No battery pack, no hidden generator, just a jar of river sludge and a completed circuit. In the previous chapter, we watched this mud come alive, powering a light-emitting diode purely through the biological exertion of bacteria living within the sludge. But seeing it work is only the first mystery. The far more compelling question is how? How does a microscopic organism buried in oxygen-free mud manage to push an electron out of its body, through the sludge, and up that wire? To survive in these environments, Geobacter has to breathe. Not with lungs, but by dumping the electrons produced during its metabolism onto external metals like iron or rust. This is a process called extracellular electron transfer. But often, the metal acceptors are too far away. The bacteria can't swim to them, so they build a bridge. When scientists first looked at these organisms under electron microscopes, they noticed something strange. The bacteria looked fuzzy. That fuzz wasn't contamination. It was a network of protein filaments extending from the cell body, reaching out into the void like biological power lines. The community had named them nanowires, but for decades the actual mechanism of how they carried electricity was one of the most heated debates in microbiology. The scientific world was split into two camps. On one side was the redox hopping theorists. They argued that biology doesn't do metal. In their view, these nanowires were like a line of people passing a bucket of water down a fire brigade. An electron would jump from one heme group to the next, losing a little energy with every leap. It was efficient enough for survival, perhaps, but it was a slow, resistance-heavy way to move power. It was the way biology had always worked. On the other side were the proponents of metallic-like conductivity. They believed these wires were behaving exactly like the copper wiring in your walls. They argued that the electrons weren't hopping. They were flowing freely in a delocalized wave, surfing along the structure with almost zero resistance. The debate raged for years. Why did it matter? Because if the hopping theory was true, microbial batteries would always be weak, limited by the slow chemistry of buckets being passed. But if the metallic theory was right, if biology could truly evolve metallic conductivity, then we were looking at a biological material that could rival the synthetics we manufacture. The answer wouldn't come from arguing over theory. It would come from looking closer than anyone had looked before. In 2019, a breakthrough study published in the journal Nature finally solved the puzzle. Researchers were able to map the atomic structure of the nanowire at near atomic resolution. What they found was hidden in the arrangement of the amino acids, the building blocks of the protein itself. Specifically, they found aromatic rings. These are flat, circular structures that stack tightly on top of one another, like a stack of coins or pancakes. In chemistry, this is known as pi-pi stacking. It's a phenomenon that allows electrons to move freely between the overlapping rings. The team discovered that Geobacter packs these aromatic rings so densely along the core of the nanowire that they create a continuous path. It was a molecular highway. The electrons weren't hopping from station to station. They were delocalized, flowing smoothly along a conductive path created by the geometry of the protein. It was, for all intents and purposes, a metal wire, but one that grew in mud. This discovery fundamentally changed our understanding of what biology can do. It wasn't just chemistry anymore, it was materials science. Nature had engineered a protein structure that rivaled the conductivity of man-made conductors. But the implications go far beyond just efficient conduction. This brings us to the most bizarre aspect of these living wires. They are part of an organism. Imagine a copper wire in your wall. If you cut it, it stays broken. The circuit is dead until you splice it back together. But a living wire heals. If a nanowire is severed, the bacterium senses the break in the circuit. It simply grows new protein filaments to bridge the gap. The system self-repairs. This capability is hard-coded into the organism's genome. The DNA contains the blueprint for the wire, the instructions for the assembly, and the logic for the repair. It is a self-assembling, self-maintaining electrical grid. We are used to electronics that are rigid, brittle, and dead. But here, we have electronics that are flexible, resilient, and alive. It represents a strange fusion of the organic and the synthetic, a true cyborg technology built not by engineers but by evolution. We now know how the wire works. We know it is a protein superhighway capable of metallic conductivity, capable of growing and healing on demand. But a wire is only useful if it connects something to something else. We have the cable. We have the power source. The question now is, what are we going to plug into it? What can we build with a material that is both alive and conductive? The answer might be larger than we ever imagined. The uranium eaters and bio-batteries. That crimson glow is more than just a parlor trick. It is the spark of a biological revolution. In our last chapter, we watched ordinary river sludge come alive, harvesting enough energy to power a light. But to understand the true magnitude of what's happening in that jar, we have to zoom out. We have to leave the safety of the laboratory and descend into the dark, toxic legacy of our nuclear age. This is a place where life struggles to take hold. It is the site of a former uranium mine. For decades, these sites have leaked heavy metals into the groundwater, a slow-motion poison spreading silently through the earth. Here, water doesn't just sustain life, it carries death. Soluble uranium moves with the flow of the water table, invisible, tasteless, and radiologically toxic. Cleaning this up is an engineer's nightmare, a problem of chemistry, physics, and staggering cost. But nature, it seems, has been preparing a cleanup crew for billions of years. Buried within the soil at these contaminated sites are a specific type of bacteria. They don't just tolerate the toxic sludge, they thrive on it. This is Geobacter, and their method of survival is nothing short of alchemy. Remember the bubble of gas we saw in the mud? That was the byproduct of respiration. We breathe oxygen to live. But in the deep, water-saturated earth, oxygen is often scarce. So, Geobacter evolved to breathe something else. In this case, they breathe the uranium itself. Through a process of reduction, the bacteria strip electrons from the organic matter around them and pass them on to the uranium. This chemical reaction transforms the soluble uranium, which moves easily with water, into an insoluble solid. It essentially turns the radioactive metal into a rock, trapping it in the soil so it can no longer spread. It is a microscopic filter, self-assembling, self-replicating, and powered only by the urge to survive. This biological containment is a breakthrough, but it brings us back to our jar of mud. If these bacteria can move electrons to lock away radioactive waste, where else are those electrons going? That red light on our desk is proof that they don't just dump electrons into the environment. If we provide them with a bridge, a conductive path, they will send their electrons across it. We call this technology the microbial fuel cell, or MFC. It is a device that harnesses the metabolic energy of microbes. In its simplest form, it consists of two chambers: the anode, buried in the bacteria-rich sludge, and the cathode, exposed to the air. The bacteria eat the fuel in the mud, sugars, acetates, waste products, and in the process of digesting it, they release electrons. Normally, those electrons would be passed to natural minerals in the soil, but we've given them a shortcut. Thousands of Geobacter cells colonize the surface of the anode, linking together in a dense, conductive biofilm. They hold hands, electrically speaking, forming a living power cable. They aren't just swimming in the mud, they are plugging themselves into the grid. As they digest the organic matter, they pump electrons directly into the graphite rod. Those electrons flow up the wire through our LED and combine with oxygen at the surface. The light shines and the bacteria get to keep breathing. It is a symbiosis between human engineering and ancient biology. The implications of this are profound. Imagine a network of environmental sensors deployed in a remote rainforest or the depths of the ocean. Normally, you'd need to replace their batteries every few months, a costly and logistical nightmare. But with microbial fuel cells, you don't need to carry battery packs. You just need the mud. Scientists are even discovering that nature beat us to the punch. Deep within the Earth's crust, vast networks of conductive minerals and bacterial cables are connecting ecosystems over kilometers. These are geobatteries, natural fuel cells where one part of the ecosystem produces food and another consumes it, linked by the slow, steady hum of electrical current through the ground. But, as incredible as these mud batteries are, we are only scratching the surface of this electric life. To truly understand the boundary of what is possible, a team of researchers decided to push these organisms to their absolute limit. They wanted to know if they needed the mud at all. This is the experiment of electrotrophy. It sounds like science fiction, but the question was simple. Can a living organism survive on pure electricity alone? In this chamber, there are no organic molecules to eat. There is no sunlight for photosynthesis. The only thing provided is a voltage, a steady stream of electrons at the surface of the electrode. The results were astonishing. The bacteria didn't just survive, they thrived. They bypassed the need to eat physical food entirely. Instead, they absorbed electrons directly from the rock to power their metabolism. They were eating electrons the same way we eat a loaf of bread. This challenges the very definition of what it means to be alive. We tend to think of life as something that consumes matter, plants eating light, animals eating plants. But here is a life form that consumes energy in its rawest form. It suggests that the biosphere is far more electric than we ever imagined. If life on Earth can exist on nothing but electricity and rock, the odds of finding life elsewhere in the solar system change dramatically. Look at Mars. It's a frozen, radioactive desert. The surface is bathed in lethal UV radiation, but deep underground, there might be water. There are minerals. There is heat. If a microbe like Geobacter can survive in a uranium mine on Earth, it could certainly survive in the basaltic rock of Mars. We may not need to look for little green men, we may need to look for little electric rocks. Or consider Europa, the icy moon of Jupiter. Beneath its frozen crust lies a global ocean, cut off from the sun for billions of years. In that eternal darkness, the energy to sustain life wouldn't come from photosynthesis. It would come from chemosynthesis, life powered by the chemical and electrical gradients between the moon's core and its ocean. These aren't just curiosities in a jar, they are pioneers. They are showing us that the requirements for life are far more flexible than we thought. Life doesn't need to be pretty. It doesn't need to be warm. It doesn't even need to eat. It just needs a circuit to complete. But if we can teach them to eat our waste and power our homes, can we also program them to build our technology? In the next chapter, we will explore the world of living electronics and the dawn of the bacterial computer. We have reached the end of tonight's topics, and it is time to let your mind completely unwind. Whatever you learned tonight, let it fade into a soft hum as you drift off. Thank you for being a part of Night Shift Knowledge and sharing this quiet space with me. Remember to leave a topic suggestion for next time if you are still awake, but for now, just rest. Sleep well, and I will be here the next time you need to quiet your mind. Good night.